chromatography section. And just like last week's activity, there's a lesson plan template for you to use if you do this with your students. Description, vocab, objectives. And then you get to 4 practice problems. And I'm going to go through how to do the calculation, but we're going to just do the calculation for the actual lab, which makes more sense to me. We can certainly go over any of those 4 that you would like to, but they're identical to what we're going to do for lab, so we can actually do the lab. So what you need to find would be your Kool-Aid chromatography bag. And I know Megan had sent out just to have everything ready in advance, but sometimes that's difficult. So we have 2 different flavors of Kool-Aid. We have a Luer lock syringe. And then we have a C18 pack. And what I have is a couple things to look at. Because to me, this is very, very interesting. And I think it's important. And I know we have a lot of teachers on a lot of different levels. So this could be AP Chem, AP Bio, Bio. You know, most classes you talk a little bit about polarity. In some context, you know, whether it's cohesion, adhesion, hydrogen bonding. So I found this little schematic showing the C18 pack. And so the C18 pack has inside of it, you can see the white column. And in that white column is a silica membrane. But attached to the silica membrane are these just giant, the 18 massive hydrocarbon chains. And so we know that hydrocarbons are extremely nonpolar. So the C18 pack is nonpolar. So that's important to understand. Because like I said last week, there's a lot of protocols we may do with different membrane for extraction, for example, purification of plasmid or DNA, where we're using a pure silica column, which is very hydrophilic or polar. Okay, these are hydrophobic, non-polar. And we are going to be making some Kool-Aid. And we're going to be pushing the Kool-Aid through this chromatography column. And what we're interested in is looking at the blue food coloring, the blue 1, and the red 40, which are located in the grape Kool-Aid. And that's the Kool-Aid that I'm going to do. You have both Kool-Aids that you can do, and I'll explain a little bit later about that. And you might choose to do this one instead of the grape. It's completely up to you. But when I was preparing these, I specifically got the red and blue. And I'm not sure if they're how well they show up, but you can see both structures are loaded with carbons and hydrocarbons. Okay, so that's the red there on your right. And if I slide it over here, this is the blue. and the blue has even more hydrocarbons than the red did. So we might predict that the blue is more hydrophobic than the red based on their structure. So this is something that you can discuss with students. You certainly don't have to. It depends on what level students you're doing this with. But if you have not done this ever, you're in for a treat, because this is a really good one. And if you choose to implement this with your students, they will send you these C18 packs, enough for your whole class, and they're going to let you keep them, which is awesome, because these are completely reusable year after year after year, which is pretty sweet. So I know we have a lot of labs ahead of us between now and late January, early February, but this one is definitely worth considering doing with your class. So what we're going to do is we are going to prepare two solutions that have varying polarities. We're going to take our 70% isopropanol that you should have, and we're going to make a 5% and a 25% isopropanol. Now, in order to do that, we need to do a little math. And this is the math I alluded to a little bit ago with those 4 practice problems. But I don't want to do those. I want to actually do the problems to make our solutions this evening. So if you have not seen, you've all seen this, but it may have been a long time ago. But the formula is C1 V1 equals C2 V2, where C stands for concentration. Concentration could be percentage, as we're dealing with this evening. It could also be a little, let's see, in 2 weeks, we'll be making gels for gel electrophoresis, and we'll be taking 50x TAE and diluting it to 1x TAE. So we'll use the exact same formula. And concentration 1, depending on how you set it up, I'm going to say is 70%. Volume 1, well, that's what we're trying to figure out. How much of this 70% alcohol do we need to add to water to make a 5% solution? So I'm going to say V1. Is that showing up okay? My Sharpie's dying. All right, good. Equals C2. Well, C2 is the concentration that we desire to make. And one of them we need this evening is a 5%. And then V2 is the volume that we want to make. And I'm just doing 50 milliliters. So you choose how much you need. 50 milliliters is more than enough for what we're doing. And then so solving for V1, we get 3.6 milliliters, dividing both sides by 70% to solve that. So this means that we want to take 3.6 liters and we want to add, we're basically going to be adding 46.4 milliliters of water. But what we really just need to do is get the 3.6 milliliters, and you should have a small 10 milliliter graduated cylinder in your box. I think she still sends those out. Okay, perfect. So you're going to get that and measure about 3.6 milliliters of the 70%. Is that what came in your box, or did she send 91? 70%? Okay. So I get about 3.6. This is super forgiving, like um in the winter when we're doing PCR, it's you need to have these exact reagents, but I'm just going to eyeball it, 3.6, and then I'm personally just going to add tap water. Okay. Your protocol says distilled water. If you have distilled water, great. But I know from doing this, tap water works fine. So I'm going to then measure, or excuse me, I'm going to then add water up to 50 milliliters, and then I'm good to go. So it's not like I need to go measure 46.4 and 3.6, right? I just need my 3.6, and then I'm going to fill it up to a final volume of 50. Good opportunity to remind your students of the meniscus. Good opportunity maybe to talk about cohesion and adhesion that causes the meniscus. So yeah, you want to make that first? You guys want to start with the 5%?
Mark, the 3.6 ml, is it alcohol? I'm going to add alcohol 3.6.
Yes. Yes, you're going to add 3.6 milliliters of your 70% isopropanol, which is our concentrated solution C1. Okay.
Mark, the 5%, that's how much we're using? The
Yes.
5%
And,
of that second concentration?
yes, we're going to make a 5% and then a 25%, and we're going to use them in the chromatography lab. Okay, so if you look at those 4 practice problems in your binder, you'll see, I mean, I think some of them even are what we're doing, except the volumes. Like the first question 3 I'm looking at is make 250 milliliters of 25%. Well, we don't need that. As it is, 50 is, we're going to be dumping some down the drain. So feel free to adjust that. So we're going to make 2 different solutions. I guess I can go ahead. Well, let's just make the 5% first real quick. Okay. I have to step away and get my little graduated cylinder. All right. You have a couple, maybe 2 or 3 or 4 plastic beakers in your box as well, if you want to use those or however you want to do it. I'm going to add my 3.6 into a 100 milliliter graduated cylinder, step over to the sink and fill it up to 50. Another great opportunity to talk to your students about labeling. And if I'm going too fast, let me know. How we doing? It's hard to tell. Thumbs up? All right. Then the other solution we're using is 25%. So we're going to do our calculation again. C1, V1, C2, V2. Where C1, I'm calling that the concentrated alcohol, 70%. V1, that's what we're trying to figure out. C2, this time it's 25%. And V2, I have just chosen 50 milliliters. And solving this, we get V1 equals 18 milliliters of 70%. So I'm going to go do the exact same thing. I am going to grab my, actually this one, I'm just going to make it all in the 100 milliliter graduated cylinder. I'm going to get 18 milliliters. Step over to the sink and fill it up to 50, and I'm good to go. And I don't want to go too fast and leave anyone behind, but you also have to let me know if we're good to go, because I have no way of knowing. Okay. While we're waiting, you can decide whether you're doing which Kool-Aid flavor you're doing. Your instructions tell you, or you know, whenever you're going back through this, but they're right on the page under preparation. That's what we're working on right now for Kool-Aid chromatography. To get 3/10 of a gram of Kool-Aid and add it to 100 milliliters of water. Now, if you don't have a scale or whatever, you're at home and you're doing this, it doesn't matter. Look at this. Just that's 3/10. But just grab a little bit of Kool-Aid and dissolve it in the water. No sugar or anything like that's needed, just the powder. And again, I'm doing the grape. So I'm going to go grab another beaker. Grab my Kool-Aid. All right, there's my Kool-Aid. This is a pretty quiet group, huh? All right. So right now,
So
go
I'm
ahead.
sorry, Mark, I had a question. What is all that you have on your board is distracting me in the back? Is
Oh,
that chemistry?
it's chemistry and physics.
Oh my goodness. Okay, I was just wondering.
Yeah, it was a good day today. Um,
Okay.
so we now have a 25%, we have a 5% alcohol, we have Kool-Aid, we still have our 70% concentrated, which we will need, and then again, a little more water for the pre-treatment of the column. The other part of this lab that's incredible is you'll see when we're done with it, it is open to so much inquiry to allow students to explore. Because it's like, you know, you don't have to do 25 and 5. What if a student were to design an experiment to test um, the different concentrations of alcohol pushed through the column? You know, it doesn't have to be, doesn't even need to be a cookie color or cookie cutter kind of lab. You can open this up with CER. You can just pose one big question and have the students, you know, explore that question. But I think that'll be more evident after we actually go through the lab together. So alcohol is nonpolar. And depending on what level you teach, you know, you can talk about the different degrees of hydrophobicity. You know, it's not necessarily this is polar, this is nonpolar, that's the way it is, because these larger molecules have areas that are hydrophilic and areas that are hydrophobic and may mostly be hydrophobic and mostly be nonpolar. Which I could definitely see in those diagrams I showed you of the red and the blue. If you look closely at them, you can see there are some regions that would be slightly polar. So alcohol, hydrophobic, alcohol, non-polar. Now, what we changed was we added to the 5%, we added quite a bit more water, which is polar, i.e., hydrophilic. So our 70% alcohol is the most hydrophobic. 25% would be the next most hydrophobic or nonpolar. And then our 5% would be our least nonpolar, least hydrophobic. And we already looked at those, the structure of the red and blue food coloring, and we talked about the structure being overall being nonpolar. And there used to be pictures of those in the binder, but those have been taken out. That's why I printed those that I found online this morning. So this column is hydrophobic, the C18. So we're going to take and we're going to push through Kool-Aid. Now we're not doing that yet, but that's this kind of big picture where we're going. We're going to push Kool-Aid through the column, and we would predict that hydrophobic biomolecule or molecules will cling to the nonpolar hydrophobic C18 pack. We would predict that the hydrophilic polar substances would elute into the trays that we're going to be using here. So you need one of these trays and you can take the lid off. And at this time, I am on procedure step 1. This is a one-way flow. So you will not be drawing up fluids through the C18 pack. Anytime we're drawing up fluids, we're going to remove the C18 pack, draw in the fluid, replace the chromatography, the C18 pack, and then slowly expel the fluid through the column. Now, before we can do anything with Kool-Aid, we have a couple steps to pre-treat. Step one is we need to push 70% alcohol through the column. So these are pre-treatment steps. Again, I'm on step one under procedure. So I'm going to do that now. I'm going to get a little extra tap water. And I sure hope you would stop me if anyone's lost or if I'm going too quick. First things first, the alcohol, pushing it through. So you can see under my camera, I do not have the lure lock on. I do not have the C18 on. I'm going to take in my 10. Milliliters. I'm going to replace the column. And then using a steady, constant pressure, I'm going to expel. It's hard to see because it's clear, but I'm pushing the 70% through the column. And it does take considerable, quite a bit of pressure to do that. And under number one, you read through it, it tells you exactly what I just went through. And so I've now expelled the 70% alcohol, which is the most hydrophobic, most non-polar solution we're working with this evening, through the column. And now I'm going to do water. It says distilled or deionized water. I'm going to save that for other things and just use tap water. So same deal. This is my water. Notice I've removed the C18. Grab about 10 milliliters of water. And now I'm going to slowly expel the water back into whatever vessel of your choice. Doesn't even matter. All right.
Are you doing that to keep everything sterile?
No, it's just like a pre-treatment, the way you pre-treat the filter to get it ready for the actual separation of the dyes.
Oh, okay.
Yes. So you do the alcohol, then you do the water.
Right.
Remember the water, very very polar, very hydrophilic.
Mm-hmm.
And any of the substances that are in this Kool-Aid that are polar would readily elute and would pass through into whatever vessel you're expelling this into. But now we're ready for the 6-well microplate. I'm on step 3 if you're following along in your binder. Step 3 under procedure, which is on the back side. Now here comes the good stuff. Time for the Kool-Aid. I'm gonna remove. And I'm saying this because you know what the students are going to do, right? They're going to stick this in the fluid and start trying to bring the liquid up through. So you really have to reinforce that this is a one-way column. So I've taken it off. I'm now going to grab 10 milliliters of Kool-Aid. Is anyone doing lemon lime out there? Lemon lime. Oh, sweet. That'll be wonderful. All right, there it is. Column back on. And now we're going to, I want to give you a heads up as you move on to step 4 here, you're going to elute into the microplate. And once one section's full, you can just kind of move on to the next one until you've pushed all 10 milliliters through. All right. So I'm going to do mine real quick here. I'm going to pause, just kind of show you mine. You see the purplish stuff that's now in the C18. It's getting clinging to our very hydrophobic stationary phase of the chromatography. And what's eluting is as clear as water from my first plate there. Look at that. Wow. Move on, that plate's, or that one's full. I move on to the next well. The intensity of the color clinging to the column has increased. So cool. Still just looks like water coming out. You know, it's crazy if the students mess this up. All they have to do is push a very concentrated alcohol through and it resets it and they can just start over. Which we'll see at the end because we're going to clean these and get them ready for storage. Okay, so in my column, hopefully yours, if you did let me rotate that bad boy. There we go. You can see the red and blue. So that is on the C18 column. Again, C18, very hydrophobic. These two dyes have varying degrees of hydrophobicity, but they are hydrophobic as well. And now we're ready to move on to step 5, which is our 5% alcohol. And of course, I always talk to my students a ton about, making predictions. Okay. I know alcohol is hydrophobic, but this is diluted all the way to 5%. So it's not going to be nearly as hydrophobic as my 25 or 70. So if there are any molecules that have a stronger affinity for the 5%, and if you want, you can do the whole like dissolves like thing. I don't do that much because I talk more about the intermolecular forces because that's what is causing these polar to dissolve in polar and non-polar and non-polar. So I talk a lot about hydrogen bonds and London dispersion forces and things like that. But here we go. I am going to now push 5% through. And if one of these molecules has a greater binding affinity for the 5%, it will elute out into the spotting well. Behold. Now I know you guys are all doing this too, so it's not like you're not seeing this, but man, that's awesome. Red. Now recall the structures I showed you at the start of this. The red was the one, it was a smaller molecule. It didn't have as many hydrocarbons associated with its structure. So we predicted that it would be not as hydrophobic as the blue. So the blue is more hydrophobic. It's more nonpolar, and it has a greater binding affinity for the highly nonpolar C18 column. So it says, hey, I'm not letting go of this column. I'm not going to elute into solution. I'm going to cling to the column. And the red says, I'm out of here. I have a greater binding affinity for this less polar substance, less non-polar, I guess. And there's my series of 3. And making observations about the column itself. I see some residual red in there. See a lot of blue. So now the question is, what if I push 25% through this? 25% is definitely more nonpolar than the 5%. And I know that this blue must be more nonpolar or more hydrophobic than the red dye. So I wonder if it will elute when I push 25% through. Now, given that this lab and the way this lab is set up and how we're following it, you're probably thinking, I think it's going to elute. But let's find out, because this is what the students could explore. What threshold, what percentage will this blue elute? You know, that to me, that's interesting. Does it have to be 25? Would it elute at 15? Would it elute at 20? All right, so I'm going to take off my column, go into my 25. You guys are probably all already done with this, aren't you? And I'm being so slow.
No, I'm learning so much. I love this. I'm nerding out over here. Thank you. And I didn't know
Why
any of
is
this
he
stuff,
so
so I'm learning
slow?
so much.
Okay, good. Man, this guy's slow. All right.
How are you spelling the loop? Because this is all new to me also.
E-L-U-T-E.
E-L-U-T-E. Okay.
And it can also be called the flow through. The flow through. All right.
I appreciate you going slow because I'm having to work from home still. And so I'm running back and forth. So
Oh, okay. Yeah,
We're-
that would be challenging for sure.
we're still at home because of the biolab fire. So
Mm.
Oh, we used to still do this training after STEM Forum. And my wife, we would get home, from Athens, and we'd have to do electrophoresis or something in the kitchen. But I guess, with everything going on this year, there it comes. Look at that. I don't know if you saw, but it was the red came out and now it's the blue. Which means the blue now has a greater binding affinity. You can throw out the like dissolves, like whatever, for the 25%. And it says, I'm out of here, C18 pack. I like this 25% better, and it elutes or flows through the column. Man, this. Now, my wife says she likes the lemon lime better, so whoever's doing that, we're going to have to check it out. Well, we have green, yellow. I mean, think of all the different things you could do this with for real. All the different, I mean, it's limitless. All right. So cool. So if I look at this now, there's a little hint of color, a little bit of blue in there, but don't be alarmed. Remember what we can do now? We can push through our 70%. And everyone will need to do this. This is how you prep it for next time. And, you know, the students used and abused on these things. If you can't get a column clean, then go get the 91% isopropanol. And that'll most of the time take care of it. So I am going to get a little bit of this 70%. And let's see. Okay, on the next page, maintenance information. Columns 100% reusable, but you have to make sure you push your 10 mils of 70 through. So right now I'm getting 70, which is the most hydrophobic solution we're dealing with. If you have 91, that's wonderful. And I'm gonna push the 70 through. And that should clean the column. So if you haven't done this, these are a must-have for the simple fact that they're reusable. I mean, I'm talking years. My wife had some, we were pushing 99% through to get them cleaned. Oh yeah. Just like new. Look at that. Boom. Chuck them in a container for next semester. I mean, it's really that easy.
So the C18 is where you can see the layers of the different colors depending on the hydrophobicity.
That's correct.
Okay.
And so the key, you know, is making sure, because even in this write-up, I don't know if there's anywhere where you are explicitly told the C18 column is hydrophobic.
Hey, can I ask a question too?
Yes.
Would this be appropriate, this column be appropriate to use for like plant pigments? Like if I could you separate like different kinds of plant pigments with these?
Oh, I'm sure you could.
Oh, that's a good idea.
Absolutely. The only thing you'd want to make sure, be careful of would be being able to clean it after it, you know, when it's done.
Mm-hmm.
But I don't see why not. And let's see, comment. I saw somewhere. Oh, amphipathic. So be both polar and non-polar. And absolutely. So that's where I would be talking about these alcohols. Maybe put a simple structure on the board, like, you know, methanol or ethanol, and talk about the fact that it has this hydrocarbon chain, but then it also has this OH group on it. So it has a polar region and it has non-polar regions. So that is super important to get into with students, because they are just convinced that it's either polar or non-polar, and that's the way it is. There's no in between. So it's really cool to talk about the structure, the more complex ones, and say, hey, this region is capable of hydrogen bonding, And this whole region here is all hydrocarbons with London dispersion forces, so it's going to be nonpolar. So let's see. Can somebody please show us the green, the lemon lime? I want to see if it's really better than, because my wife thinks it's the best. I think they're both cool. Obviously, you could do them both.
Oh, the lemon lime is pretty cool looking.
Okay.
I
And
like the purple better though.
see, that's what I'm saying. And what is neat though is if you do it both, you can then pull up the structure for the yellow dye, and you can see that it would be the least hydrophobic of the three dyes. And so again, I wonder what threshold of alcohol, like what percentage would you need to get that yellow to elute? Those are just things that I love having my students explore. So they're all kind of doing different things when they're doing the lab. So
why would the industry want to do this method?
So chromatography is used to purify so many substances like pharmaceuticals, medicines are purified through. If you look at that's where this lesson plate template, if you go back to that under applications.
Mm-hmm.
So food industry, perfume manufacturing, chemical purification.
That's in our binder.
Yes, it's in your binder at the start. It's the lesson plan template for this. And each lab we do will have one of those lesson plan templates.
Okay, I'll go back and look through it.
And, I know for my research class, well, a bunch of you have probably purified plasmids from Piglo, maybe, any of the biotech teachers. So those mini preps are silica chromatography, and it's all the same idea, except you're taking your plasmid and you're purifying it from all the other cell debris. And the plasmid, let's just say the DNA clings to the silica column because it's very hydrophilic, and it clings, and throughout the process you're pushing all of these different reagents through the mini prep, except you're not pushing it with a syringe. You're doing it with a vacuum or a centrifuge, but you're forcing the liquid through the column. And as you put different reagents in, they become more and more hydrophilic. And so kind of like the opposite of what we did this evening. And eventually you're getting rid of all your flow through, and you're throwing it away because it's all the cell debris that you do not want. And then at the end, you can push your most hydrophilic solution in. A lot of the kits give them these fancy names, super fancy proprietary names, but it's either TE or water or something super polar, and the DNA will elute and that's when you have your purified DNA extraction.
So is that similar to how the centrifuge works when you have the little pellet at the bottom?
Yes, that is exactly right. Yeah, well, the centrifuge, you're forcing the fluid through what they call a mini prep.
Mm-hmm.
But it's basically, it looks kind of like this, except you set it into a microcentrifuge tube, and then you use centrifugation to force the fluids through the column.
Mm-hmm.
So a lot of different applications for chromatography, tons of applications.
Mark,
Yes.
I also teach research. Have you used this in any of your research projects when they wanted to isolate something?
The C18, I have not.
Okay. I'm just
No.
thinking about what I could use it maybe to separate and then run it through like the SpectraViz, you know.
Yeah, I'm sure you could. I'm trying to think. I don't recall students ever using it. But we are always using the plasmid and the DNA QIAGEN DNA blood and tissue kit for extraction and other kits. I guess they're all very similar for environmental DNA, soil, plants, all have some sort of chromatography in the extraction.
Okay.
So,
Thank you.
you're welcome. That's awesome. Anything else? Anybody questions or thoughts? I think I've thrown out a lot of different ideas for extensions for using these. Would just be exploring CER, put a question on the board and let them do their thing. You know, have them run their plan by you, of course, because we don't want to be destroying the columns.
Mark, I have a question.
Yes.
All right, can you go back over and tell me again, I'm a little bit slow here, why the isopropyl alcohol is hydrophobic?
So the alcohol does have hydrophilic regions, right? But
Right.
overall, it's hydrophobic. It's non-polar. Now the CPAC is next level non-polar, next level hydrophobic, full of just hydrocarbon chains. Whereas these alcohols are going to have those those OH groups on the ends,
Mm-hmm.
which are regions where that would be hydrophilic. But overall, it is hydrophobic due to the hydrocarbon chains, which
Okay.
are extremely hydrophobic, have that very low electronegativity difference, and will form the London dispersion forces with other hydrophobic regions.
Okay.
Okay. Does that
That
help?
helps. Yeah.
Okay. Anything else? Anybody else use these and have ideas that they want to share? It is 6 o'clock. We are on till 6:15, but that's everything for our training this evening. So
So is a class set of these expensive?
does anyone know? I feel like these are about $6.
Oh, wow. Okay.
I could be wrong on that.
For one?
Pricey, but
Yes, but remember, reusable. See, I don't even remember because it's been so long since we bought these.
I think I paid like $13 from Bio-Rad.
13 from
Yeah.
Bio-Rad.
I was actually going to fuss at you for gatekeeping because when I looked these up, you could get I think like a 50 pack for 190, which was actually cheaper than what Bio-Rad does, but they do a smaller amount.
And also, you guys, we may want to get with Megan because there was an industry partner that I think can get these even cheaper than Bio-Rad. Back in the day, we used to get them from Bio-Rad. Let me step over and grab the card. I think I have it right here. Yes. And it is Zorb Tech. I'm gonna put it in the chat so you can see it because of spelling. We want to just remember to ask her about this. Okay, that's what I'm doing. Because if she knows, and that's where I'm getting that cheaper price in my brain was from this. And they used to be a big partner at Georgia Bio. I'm sure they still are. And they specialize in chromatography. So we need to ask her about that. I feel like this is a different C18 than Biorad cells.
Yes,
In
it's
fact,
much smaller.
okay. So, but we can ask her about that and get a price point.
Hey, Mark, this is Mel with Georgia Bio. I'm happy to shoot Megan a note about it and get y'all an answer on pricing.
Oh, thank you.
Of course.
That'll be great.
Little voice of God over here.
Thank you. Anybody else? Any thoughts? Okay. Well, I mean, I will certainly stick around. I hope you enjoyed the labs this evening. I know I sure as heck did. And if you're good to go, you can sign off straight from the breakout room, and I'll see you in 2 weeks. And thanks for hanging out with me tonight. That was fun. And I'll stick around.
Thank you.
You're welcome.
Thank you.
Thank
You're
you.
welcome. I can hear